Nano-enabled immunotherapy and cancer. Ladies and gentlemen, it's an honor for us to have André with us. He is a professor, distinguished professor of medicine, chief and division of the Nano-medicines research, director of the California NanoSystems Institute, and director of the UCLA Center for Environmental Impact of Nanotechnology, and he is also the associate editor of ACS in Los Angeles. He is a specialist in the field and has a long experience in the field of nanomedicine. May I ask you for your talk, André? Thank you very much that you have accepted to come.
Thank you, Beat, and thanks for the invitation under unusual circumstances. I hope everybody is safe. So I'll be talking about nano-enabled immunotherapy for cancer and the treatment for allergic and autoimmune disease. So I'll just summarize that the significance of what I'll tell you of the two types of studies in my lab is, number one, is impacting the scope of cancer immunotherapy by what we call an endogenous vaccination approach, and that is to enhance the immunotherapy response to checkpoint inhibitors by therapy that turn cold tumors hot, so as to enhance the checkpoint response by what we call chemoimmunotherapy. The other side is tolerogenic immunotherapy for systemic allergic disorders and autoimmune disease, of which is significant is that amongst the pandemic of autoimmune disease and serious allergies, including food allergies, tolerogenic therapy can provide a safe form of therapy. The bottom right-hand side is just to show you that for autoimmunity, actually the annual US cost and the prevalence of this is much higher than cancer. But look at the bottom line, where the research funding for autoimmunity is only 1/10 the amount that we get for cancer. So the basic approach is to use nanoparticle engineering of the immune system, where we custom design nanocarriers to either provide immune stimulation or immune suppression. This can be done by using the nanocarriers to deliver medications or allergens, as I will demonstrate, or you can also use the nanocarriers shape, size, elasticity, surface charge to send signals to the immune system. For instance, I show here one of our studies where we've modified alum as an adjuvant by making it into nanorods, which provides a much stronger immune stimulus. So anyway, the use of the engineered technology allows us then to do immune stimulation through chemoimmunotherapy, and then on the side of autoimmune disease to treat antigen-specific immune suppression by making use of the biology of Tregs. Regulatory T cells, we also say here in California, if 2 Tregs walk into a bar, everybody calms down instantaneously. So first, let me take on the side of the cancer immunotherapy where we use stimulation. The idea here is to turn cold tumors hot so that we can enhance the response of immunomonotherapy by using of combination therapy. And the possibilities of doing this exercise of taking cold tumors to hot is to use vaccines and adjuvant. So what I will show you today is the use of chemotherapy as a form of endogenous vaccination. And that works on the basis that a number of chemotherapeutic agents such as anthracyclines, paclitaxel, mitoxantrone induce a form of cell death other than what chemo normally does, namely apoptosis and necrosis. I'm talking here about the generation of immunogenic cell death by a select few chemo agents, where the induction of the cell death and nuclear disintegration generates an ER stress stimulus that provides calreticulin expression on the surface of the cancer cell that provides an eat me signal to dendritic cells in the immune system. The same cell death response is also accompanied by release of a nuclear protein, HMGB1, as well as a process of autophagy that releases ATP. HMGB1 and ATP provide adjuvant stimuli to the dendritic cell to generate immune responses. The ability of chemo agents to do this can be tested by taking cancer cells, exposing them to the chemo agent. If you vaccinate the animal in one flank and then wait for a few weeks and then inject the live cancer cells in the opposite end, you can actually see the immunogenic response. And that has then looked at the characterization of a long list of chemo agents, some of which are shown here. For instance, oxaliplatin, and then some of the strongest responses being induced by doxorubicin and mitoxantrone. So the first application that I will show you for immunogenic cell death is for pancreatic cancer, where we've developed a nanocarrier system that consists of a nanoparticle that is a hollow mesoporous silica nanoparticle coated with a pegylated lipid bilayer. These nanoparticles allow very, very high loading capacity, and we can produce them in large quantities and large volumes now for preclinical studies. The mesoporous silica nanoparticle, the example that I'm going to show you, is a chemo agent that to date have not been demonstrated yet to play a role in immunogenic death, namely irinotecan, which can be imported across the lipid bilayer at very high loading capacity in these particles. You see the CryoEM with a lipid bilayer and the hollow mesoporous interior. These particles were developed as a liposome simulation technology or liposome lookalike, both with lipid bilayers, but have the advantage in the case of pancreatic cancer of containing much higher drug loading capacity, a stable lipid bilayer which is less leaky. So the circulatory half-life of these particles are longer, and their stability also allows much higher biodistribution to the pancreas tumor site and drug delivery. On the bottom left-hand side, I show you, we use a pancreatic cancer model, orthotopic implant of the pancreatic cancer of the tumor cells in the pancreas. The performance of PK studies demonstrate that compared to the liposome, to, for instance, Onavid, which delivers irinotecan, the silicosome performs much stronger in terms of drug delivery. The primary tumor grows and it metastases occurs. In the treatment of free irinotecan, very little impact on this very rigorous tumor. The irinotecan silicosome reduces the metastases somewhat, but you still see considerable amount of tumor. With the irinotecan, all the metastases are gone and there is essentially no primary tumor left. Now, what was an important recent discovery, irinotecan is a topoisomerase inhibitor, is that actually in this case, the immunogenic cell death is due to the nuclear damage, but has a primary component to it, something that happens earlier, where the irinotecan rapidly enters the lysosomal compartment, where the amines provides neutralization of protons in the lysosome. This leads to alkalinization of the lysosome. This process here is then responsible for interfering in autophagy flux, which removes a lot of cell waste and oxygen radicals. The accumulation of the oxygen radicals induces a strong form of ER stress, which then promotes the calreticulin and HMGB1 expression, as I described earlier on. This form of cell death, where ER stress plays the primary role, is also known as a type 2 immunogenic cell death response. Importantly, the immunogenic cell death response will generate the hot TME, but an important discovery was that autophagy also has a sensor system which activates a cell pathway, the NF-kappa B pathway, that expresses PD-L1 as a checkpoint that then interferes in the immunogenic cell death response. So how does this fare in the orthotopic model? Well, in addition then to the tumor cell death, we also see quite effective induction of calreticulin expression. So there is immunogenic cell death that occurs with a silicosome, but not with a free irinotecan. On the side of immunity, a whole series of events that take place, but there is generation of CD8 cytotoxic T cells, and also a highly significant increase in the ratio of cytotoxic to regulatory T cells by the irinotecan silicosome compared to free irinotecan. Also noteworthy, the new data also shows that there is effective induction of PD-L1, and therefore a study was undertaken where we used the silicosome together with anti-PD-1 antibodies. And in the survival study, the Kaplan-Meier plot shows that compared to the saline anti-PD-1 or the free irinotecan, we do see response improvement if we use the silicosome alone. But if we use the silicosome in combination with anti-PD-1 therapy, a highly significant increase in animal survival in the study. The second example to the use of the immunogenic cell death is the studies that we do on triple-negative breast cancer, which requires combination therapy with a system of liposomal carriers that we have developed to deliver dual drug delivery. And this is necessary because here, an ICD stimulus with the activation of the cytotoxic T cells produces interferon gamma, which has a negative effect of the newly generated immune response. Interferon gamma is responsible for IDO expression, indoleamine 2D3 deoxygenase, which generates Tregs that interfere. Interferon gamma also activates the JAK-STAT pathway, which in cancer cells induce PD-L1 expression. So there's also a contribution of immune interference by PD-1. Here, the approach is to combine stimuli where we use the ICD stimulus plus interference or small molecule inhibitors in the IDO and PD-L1 pathway. And I show you one type of liposome that have been developed where we use chemo agents, plus we put an inhibitor of IDO, also known as indoximod, into the lipid bilayer. And of course, all of this needs to also take into consideration that in addition to cold tumors that you want to turn hot, in these environments, even after you turn the tumor hot, some of the T cells are excluded from the immune environment by a sequestration process. So the liposome was designed then first by using a prodrug design to convert indoximod into a cholesterol prodrug by an esterification process. The prodrug could be placed into the liposomal lipid bilayer, which requires the product has a cation, put a negatively charged cholesterol hemisuccinate into the liposomal membrane. The mitoxantron, which I will use as an example here, is remotely imported by using citric acid. Here you can see the CryoEM of the liposomes, where the drug precipitate is that almost smiley face that you see inside the liposome. If you study the PK of delivery in a breast cancer model, both the single mitoxantron as well as a dual delivery particle quite effectively increases the pharmacokinetics, as for instance demonstrated here by the half-life as well as the area under the curve for the liposome. Performance of animal studies on triple-negative breast cancer models, where we use orthotopic implant with tumor growth and metastases, In both the 4T1 and the EMT6 model, mitoxantrone liposome is quite effective for shrinking the tumor and inducing an immune response, and the response was further effectively enhanced by a dual delivery liposome that also interferes in the IDO pathway. The response is accompanied by a highly significant increase in calreticulin expression, so there is an immunogenic response, stronger for the dual delivery versus the mono delivery liposome. But a big surprise was when we looked at the immunogenic event. Instead of what I showed you with irinotecan, CD8 T cells are dramatically decreased by the two liposomes. And that required exploration to look at the form of cell death. Cell death, like for cytotoxic T cells, there is still deposition of perforin, that's the staining in green, in these tumor sites. But if you go into tumor site where you do a triple color staining with its cell nuclei, the CD8 T cells and the perforin, you will see that treatment with the liposome leads to disappearance of the CD8 T cells, and yet there is deposition of perforin. Where does the perforin come from? Well, another cell type that produces perforin is NK cells, and in a triple stain with perforin and NK cells, the treatment with the liposome induces in red the NK cell appearance, highly statistically significant, with the deposition of the perforin and cell killing. So, in this part of the talk, I'm saying that in terms of the heterogeneous landscapes, now we have interesting that we have mitoxantrone that induces an NK event. I've shown you irinotecan and pancreas cancers that can induce CD8, a CTL event by a type 2 response. And then for the triple negative breast cancer, if you use doxorubicin, look what happens. Doxorubicin effectively induces, this is staining for CD8 T cells, a highly effective increase in CD8 T cell expression, but which is excluded in the margin of the tumor. Mitoxantrone, disappearance of CD8 T cells, only a few sitting in the margin, but infiltrated by NK cells. So in order to dynamically design the therapy, we're looking at combinations then of different ICD stimuli, immune escape inhibitors, IDO1, PD-L1, other checkpoint pathways, and then also intervention in T cell exclusion pathways, which I will not have time to talk about. The other end of the story is to use nanoparticles for immune suppression. And the model that I'll describe to you comes from studies on the liver, where if you transplant a kidney, you have to use a lot of immune suppression. If you transplant a kidney and a liver, very little immune suppression is needed because the liver itself is an immune privileged organ, which protects against food allergens, pathogens, by a series of locally expressed antigen-presenting cells that specializes in immune suppression. And it does so by inducing from these antigen-presenting cells regulatory T cells that can go to site of pathology. So we use this platform for the treatment of serious allergic disorders and autoimmune disease. So how does the invention work? Well, I'll show you from the microanatomy of the liver, several of the antigen-presenting cell types in the liver, which can act in an immune suppressive sense. Among those, the liver sinusoidal endothelial cell is the strongest suppressive antigen-presenting cells, which is present in the liver microanatomy, in the liver sinusoids, occupies a huge surface area. Different from, say, Kupffer cells, these cells process nanoparticles by endocytosis. And this is a process where they can use scavenger receptors that bind to ligands. And I show you two ligands, ApoB and mannan, and the scavenger receptor stabilin, and you can see the stabilin being expressed exactly in that sinusoidal fashion. If you engage the stabilin with a surface receptor, the antigen is presented by the liver endothelial cell to CD4 T cells of a specific type that induces TGF-beta, and this generates the regulatory T cells. So the prototype carrier design is to use a PLGA polymer design, which is a facile synthesis process. It's very scalable. We can use microfluidics to make it in large quantities. The polymer is FDA approved. It has high intrinsic safety, and there's the ability then to load into this carrier the antigen that you want, or an epitope of the antigen. You can also put in drugs, you can put in immunomodulators, and then very importantly, decorate the surface of these particles with an ApoB ligand or mannan to target it to LSACs. On the right-hand side, you see a scanning electron microscopy of the 200 to 300 nanometer sized particles. How well does the targeting work? Well, I'm going to show you here an antigen that causes food allergy and also severe asthma in mouse models, namely albumin. We inject albumin intravenously. Most of the albumin, which is fluorescently labeled, distributes to the kidney. Very little goes to the liver and lung. If you stain for the liver sinusoidal endothelial cells, or LSACs for short, you see the sinusoidal distribution, but there's no antigen present. If you put the albumin in a nanoparticle that is with ligands on, the majority, more than 90%, goes into the liver and redistributes in the liver with staining overlap with the LSACs. So you do get effective liver targeting. How does it work in terms of one of those animal models? Well, the animal models, we then, one approach is to use the nanoparticle to pre-treat the animal before we sensitize the animal to albumin with intraperitoneal sensitization. We expose the animal by inhalation a few weeks later to aerosolized albumin. And what then happens, the animals develop a very, very strong asthma-like immune response with Th2 cytokines, IL-4, IL-5, IL-13, and a heavy infiltration of eosinophils into the lung. It's also been shown in basic immunology studies that each of these phases involving multiple cell types can be controlled by Tregs. Okay, so what happens if we expose animals to OVA and we pre-treat them? This slide here shows the eosinophilic inflammation in the lung. Oversensitized and challenged animals develop a very strong eosinophil response. If you treat it with a nanoparticle that contains albumin only, there's no change, still a strong eosinophilic response. However, in nanoparticles, where we incorporate albumin with mannan on the cell surface or the ApoB peptide, there's a progressive decrease, so that especially for the ApoB ligand that targets the LSAC, there is no airway inflammation. This response, if you look in the bronchoalveolar lavage fluid of the lung, also shows then that that T helper 2 response is dramatically diminished by disappearance of interleukin 4, interleukin 5, again, most dominant for the ApoB delivering particles. Already told you about the eosinophils. There's also a decrease in IgE production. There's no increase in interferon gamma production, which is a T helper 1 cytokine response, but there is an effective generation of TGF-beta, which is a cytokine involved in Treg generation and especially prominent for the particles that contains the ApoB ligand. We now look in the lungs of these animals what is responsible for suppressing the response. By staining for the regulatory T cells, you will be able to see from this immunohistology stain a progressive increase in the amount of Tregs that is present in the lung, again, most prominent for the animals that contains the stabilin ligand on the particle surface. So what I've basically shown you then is the ability to then use the natural tolerogenic effects of the liver to inject particles that target the liver presented by LSACs for generation of a T cell response that favors production of regulatory T cells which go to the site of pathology to suppress the immune event. We can also do the same experiment where the tolerogenic nanoparticles are delivered post-treatment. That means after you sensitize the animals, you deliver the particles. And again, if we follow the allergic airway inflammation, the strong eosinophilic inflammation that develops in the lung can be reduced on post-treatment of albumin nanoparticles, which is especially prominent if you put surface ligands on, and total disappearance of the response in case of nanoparticles that's decorated with ApoB. I'll just say that in these animals, the IgE response does not disappear because once B cell class switching has taken place, you cannot easily switch it off. Of albumin is immunogenic by virtue of different T cell epitopes that is presented in the one case, an OT2 peptide is presented by MHC2. That's the type of MHC complex that generates regulatory T cells. Cytotoxic T cells are activated by OT1 peptide. So we also made a series of particles then that incorporates OT2, OT1, and of albumin. And using in the pre-treatment model, we could show that the MHC2 OT2 peptide is as effective as the whole of albumin in suppressing the eosinophil response, and even better than the of albumin in suppressing the IgE response. So the technology can therefore also be practiced by using epitopes to suppress the disease event. Finally, I'm also going to show you that if you use the same of albumin in an animal model where you sensitize by injecting it intraperitoneally and then challenging, that you can induce an anaphylaxis response, where there's a drop in body temperature, the animals develop a shock-like state, there's huge mast cell release that is pre-stages this, and in a pre-treatment model with both the OVA or the OT2 peptide, we can suppress the anaphylaxis response, but not with OT1. As for the asthma response or the allergic airway response, the response is suppressed by TGF-beta that generates regulatory T cells. Okay, so I'm going to conclude there to say then that for suppression, we've developed a tolerogenic nanoparticle system that makes use of the natural tolerogenic effects of the liver to provide a generation of regulatory T cells. There are also other technologies, for instance, Selecta Pharmaceuticals, who generated a nanoparticle delivering rapamycin plus your antigen. They do not select for a specific type of antigen-presenting cells, but the pharma agent locks the antigen-presenting cells into a tolerogenic state, so that there is generation of FOXP3. Of course, all studies require an excellent team, and so I want to acknowledge my junior faculty, Wan Meng, Chan Jia, our postdoctoral fellows, our staff scientists, and our students in their contribution to the work, as well as my colleagues in the UCLA multidisciplinary pancreas cancer team. Finally, this work is all done in the California NanoSystems Institute, where as the research director, we have an amazing team where we aim at doing basic discovery into translational research and commercialization with a view to leverage investment by revenue generation. And we also have our own incubator on the UCLA campus, in which there's 35 companies incubating. And then through the team science efforts that we are able to generate a steady stream of federal income where we are very successful in proposal and submission. Thank you very much. I'll be happy to take questions.
Thank you very much for this nice talk. And we have not many questions. One of the questions was regarding the targeting in the combination therapy. How would you specify the field of targeting regarding dying only cancer cells?
Okay, you're talking about the not the liver targeting platform that I spoke about, but you're talking about in the immunotherapy of cancer studies. Does targeting of cancer cells is that successful? Well, as you know, it's a complicated area of study in the sense that targeting should make a lot of sense for cancer. But the problem is that targeting only comes into play once the nanoparticle has effectively entered the cancer site. And the ability to do so by targeting design of nanoparticles, various successes have been demonstrated. But there is, at the moment, no clear one cancer that I can think of where the exclusive use of a targeting ligand has provided the sole basis of a successful outcome. What is important is to get those particles or carriers into the cancer site. And in that area, there's another very controversial area of the so-called enhanced permeability and retention effect that apply. I will just say briefly that at least in the case of the triple negative breast cancer, it would seem from recent publications that the major pathway for entry is a transcytosis process rather than a leaky blood vessel process. If you can use that transcytosis process effectively by targeting, and we've done that for pancreatic cancer by using RGD peptides, you can enhance the transcytosis import of the silicosome nanoparticles into the pancreas cancer site. But I would also hasten to tell you that we can do that with RGD peptides not attached to the particle surface as well, almost as effective as attaching it to the particle surface, because the mechanisms of transcytosis entry only requires opening the gate and the particle will go through. You do not necessarily, in that case, need to have the ligand on the particle surface.
There is another question from Jose Carbalido, who asks, what is the size of your particles and will your immune tolerance work in splenectomized mice?
Thymectomized mice.
Yes.
Yes. Okay. The particle size is around 200 to 240 nanometers, which is very important in the sense that the endocytosis mechanism that I showed you is a clathrin-mediated endocytosis event where the size of those endosomes is around that size range. So that's done strategically from that perspective. The ability to generate the Tregs relies on peripheral Tregs and not centralized Tregs. I have not done the experiment in thymectomized animals. That will still need to be done, but my suspicion is that the Tregs in the periphery is what is responsible for the liver tolerogenic process.
Thank you very much, André. Our time is over for this session. I am very grateful that you have come. It's a most interesting topic, and I think we will continue as soon as possible again in real life in Basel.
Thank you very much, Beat. Best to everybody.
Thanks.
Thank you.
All the best.